I have perhaps the most difficult job in introducing these stars, and thank goodness I don't have to reintroduce Johann because he has been introduced. And thanks for your amazing keynote address to kick us off. And then to my immediate left is Mark Harfensteller, and Mark is the COO of ITM, Medical Isotopes in Germany, and obviously is an incredible international leader in this area. Then we go to Ting-Yim, who is professor of medical imaging and medical biophysics and oncology at Western University, and is the director of the PET-CT program at St. Joe's, and obviously has had an incredible experience in research and translational research. Kate Vallis, well known to us here at the Princess Margaret Cancer Center, as she was a radiation oncologist before where she is now as a clinician scientist and heading the experimental radiotherapeutics group in the Department of Oncology at Oxford University. So incredible pedigree of experts that I have today, and my job, and quite frankly, I'm not even sure why I'm here. So I'm a surgeon. I'm a head and neck cancer surgeon, but I'm also a researcher. I head a large guided therapeutics laboratory that's funded by the Terry Fox Research Institute in theranostics, and I collaborate. That is the key of what I do with other research, Gang Zhang and others. But here I sit, I think, and one of James, our opening introducer, introduced me as someone who struggles, because I think I'm here as a health policy provider. I'm vice president of Ontario Health at Cancer Care Ontario. And in that capacity, my job is to listen to experts, like Ur Messer, for example, who is our provincial head of medical imaging, head of molecular imaging, and someone like others who are the provincial heads of medical imaging. So my job is to horizon scan, and my job is to think of where we're going in a health population, a health system of 15 million people. So yes, I struggle. And so part of what we want to bring out today is what's next. What's next on the clinical research side? What's next on the translational side? How do we take this to a population of people for therapy? And so this group is going to answer all of those questions today. And so I'm going to start with our first question. What do we need first from our preclinical scientists to take clinical treatment to the next level? So, Kate, you're up to bat.
Okay. So I think it's very apposite that we're starting with a question about preclinical research, because I do think that really preclinical research has really underpinned, the current explosion in radiotheranostics. And we're talking about, fundamental radiochemistry, biconjugate chemistry, as well as, peptide and protein engineering. So I think that's a good question to start with. What do we need from preclinical research going forward? Well, I think we need to think about target discovery. So, screens for new targets. What we really want, if we're honest, is, another PSMA, another SSTR. So good targets for radiotheranostics, and their identification is fundamentally important. I think we need to think beyond peptides. The current situation is very much dominated by peptides, but, given that there are potentially extremely useful antibody formats, not just whole IgGs, but other formats and small molecules that could be very important in radiotheranostics, we have to consider those. And I think we have to think about fundamental radiation biology, because so much of the current situation, so much of what we're doing clinically has been influenced by our knowledge of external beam radiotherapy. But we know fundamentally that the radiation biology of low dose rate radionuclide therapy is profoundly different. And we need to take that into account, particularly when we're thinking about new combinations, I think. So
I want to get to the combinations in a minute, but let's go to Ting next. So Ting, let's say Kate gives us the next target. So my colleague here, Elaine Mertens, who's my co-vice president of Ontario Health, Cancer Care Ontario, we've got liquid biopsies coming down the pipe. We have gene therapy. We have, of course, radiotherapeutics, theranostics. How can we, as clinicians, on the clinical side, as we translate to our patients, how do we build, I'll use the word, because I got to talk to politicians, we got to talk to funders, how do we build the case to make Kate's next best target into practice?
Well, that basically is tie into collecting data that will support the cost-effective analysis, right? So from that point on, you would have to design kind of new imaging techniques that will sensitively pick up those targets that are present in the tumor, whether it's liquid biopsy or using imaging, it doesn't matter, but we have to detect those targets. And then from that, you would have to kind of work out kind of what Kate has mentioned about the radiobiology of how the cells are being killed. And then further down the stream would be kind of looking at kind of how are you going to implement this in the healthcare system in the most cost-effective ways. So it's not just causing the hospitals or the cancer centers to treat the patient. There's also a personal cost to the patients as well. So some people has put this as not just a biological toxicity, it's the financial toxicity to the patients too. So all this is a continuum. So identifying the target is only the first step, and all those detections kind of improving the effectiveness, reducing the adverse rates, and then finally come out to a cost-effective analysis, because mostly is that we are looking at a incremental effectiveness ratio of $50,000 per quality year that you gain. So that is a benchmark.
Yeah, no, thanks. And so now I want to go to, and Johan, you commented on barriers and challenges, and Mark as well. So I want to go to our international experts. So where does all of these targets, and we're going to have more than one that Kate and others will tell us about. So where does this whole thing fit into the cancer treatment paradigm, monotherapy, polytherapies, in collaboration with radiation, of course, other systemic therapies, and of course, let's not forget the most historical and highly curative treatment of surgery. Where do you guys sit? Where does this sit in the whole paradigm of cancer treatment? Johan first, and then to Mark.
So I think the key difference is that this works like a whole body lesion biopsy, and that's probably the paradigm change that is most important. So you really see the targets, you see the heterogeneity of target expression, and then you can treat it. But there are some examples where it will not affect clinical management. Think about, you can image estrogen receptor expression, but I don't think it will really influence much how these patients will be treated, because they will be put on some inhibitor, some blocker. The same is probably true with HER2. If you see expression in one lesion but not in others, so you see expression in the primary but not in metastatic lesions, I don't think it will deter oncologists from treating. So you need to have a high confidence in in kind of reliability of your and fidelity of your assay. So if your diagnostic assay doesn't have the sensitivity, or if there's partial expression in some lesion, but there's heterogeneity in expression, I think then treatment will not be much affected. I think the key is really that you look at target expression at the whole body level. That's the difference. That was not doable before. You can't biopsy 40 metastatic prostate cancer lesion or breast cancer lesion. Can't do that. This is now visible. You can see that stuff in the high-performing theranostics. Then there are some, of course, where people get extremely happy and excited, and the diagnostic arm may be very good. I would say fibroblast activation protein can be very useful. But I think the therapeutic, based on also the slide that I show with the uptake on images, the therapeutic may not be very promising alone. And then the next question that comes in, how do you address it with combination treatments? So is there a way to exploit cancer liabilities in addition to just exposing them to radiation? Yeah?
Mark? Perhaps to add from the isotope perspective side, and more focusing on our therapies. So we have now the universe, let's say, of new peptides, modalities, everything, and you want always to combine it with the best isotope. And I think there's not a best isotope because you have also to fill a lot of requirements. You have to have it available on a day-to-day basis. It has to have the right properties for the treatment. So what is the pharmacokinetics? What is the half-life of the isotope? But also then how do you want to irradiate this target? You have a mild beta emitter, you need more energy in the beta or alpha. So also from that perspective, for the community, it's important to have a toolkit of not too many, but still a couple of alpha, beta emitters and so on for the treatment to make it available. And I think the combination of these isotopes and the different targeting moieties will have then the main impact. I think also right now, we are all very happy about Lutathera Pluvicto, having this monotherapy and really a big and great breakthrough. But still, I think the future will be way more complex, going to combination therapies and trying really how best you fit then and this new treating modality and the overall treatment scheme for the patients.
I want to step back a bit more and get away from targets, unless Ting or Kate, do you have any further to add to that question?
Well, the only thing I'd add, I think, is that as Johannes has indicated, is that there is a great deal of heterogeneity that we have to deal with. And so essentially, although the molecular target might be, a single target, we are trying to deliver enough radioactivity even to lesions that have low expression levels of the target. So that really is a major challenge, I think, because, within the same patient, we'll have lesions which are highly avid and other lesions which are less so. And so I think that's a real challenge for us. And, maybe one way is to come up with sensitizers so that, for the same amount of radioactivity, you get a bigger bang for your buck. And specifically sensitizers to lesions where there is low accumulation of the radioactivity. So, there's a lot of work to do.
And I also think you could add one thing that is not done yet, and that's mostly because coverage is a problem. Because people think, for instance, PET imaging is expensive, which is a bad joke. Because when you look at healthcare cost overall, PET imaging is less than 1% cancer care costs. It's rigged, what the former president say, I think. But we could phenotype cancers, and we could use not only PSMA in some cancers, we can use additional bombazine imaging. We could even use somatostatin receptor targeted imaging in some prostate cancers to find out whether they actually would be potentially benefiting from different therapeutic approaches or combined radioligand or radiopharmaceutical approaches. And I think we haven't even scratched the surface on this, and combination treatments, and what you said, optimizing, isotope half-life, retention time, on-target issues, and so on and so forth. So I think we're in the beginning of this whole thing.
We're in the beginning of this whole thing. I think it's a really good statement. So I want to, again, as I said a minute ago, I want to step back and get to barriers and challenges. And one of the things that I've learned in my role as a researcher here in collaboration is so key, and is not to label people. And so much of our success here is going to be who is a surgeon anymore when we're talking about radiotherapeutics? Who is an imager? Who is a radiation oncologist? The concept that we have here is that the fuzziness between roles becomes kind of unclear, right? And so how do we maintain respect of our respective disciplines and the expertise and yet move this forward? Because that's absolutely key. So when we prepped for this panel, we talked about ego. We talked about discipline ego. We talked about institutional ego. So you talked to Johan about some of the barriers and challenges, but I want to now explore this concept of collaboration and really the sum being greater than its parts by that. So maybe I'll start with you, Kate, go to Ting, and then I'll go all the way down the line here.
Yes. So, I think you could argue that radiotheranostics is kind of the poster child for multidisciplinarity, right? It absolutely depends on chemists and radiation oncologists and nuclear medicine physicians and imaging scientists, the whole lot. So I think we have to sort of think beyond those titles and, focus on people's individual skills and what they can bring to the problems we face. I've heard it suggested, and actually Rebecca sort of mentioned this in her introductory talks, that we should think of nuclear oncology as a new major pillar within oncology. And that, to me, seems quite a sensible approach, because it's not about a particular discipline, it's about a particular form of treatment. So those are my thoughts on the matter.
Point. Ting, over to you, and then...
Yeah, I just want to add on to that. Radiotheranostics is a whole continuum, as Kay has said. It goes from the bench to the bedside of the patients. So communications between the different professions involved is really important, and kind of workshops and symposiums like this that involve kind of different clinical disciplines together with industry, so that we would kind of identify some targets that every one of us would feel that we are contributing to it, so that we will be able to kind of work collaboratively and also understand each other's language. So say, for example, I'm a medical physicist, so sometimes I talk in terms that is completely kind of out of the ordinary. So it's this type of finding a common language so that we could communicate effectively and also collaboratively between the different disciplines is really important. And maybe there is also the professional societies would also have a role to play in here. For example, the surgical oncology and the nuclear medicine, and then also the clinical people that they could come together and come up with a kind of a guidelines as to how these collaborations would work kind of peacefully. Quote. Yeah.
And I think this type of summit here today is actually a great step in doing some of these things around breaking down silos. Johannes, you head up a large center. How have you been able to do this in a practical
So I think when it comes to egos, and since the good old hippie days are long gone and there's no LSD that pulverizes the ego, it is worrisome that everybody agrees on teamwork and collaboration, and we're all in it together. But the question of leadership is interesting because there's group dynamics. Someone will be the bully. Someone will be the strongest politically. Someone will have some other features that make him or her a leader. And that does not always jive with the concept of collaboration. It's often territorial, not by fields, but territorial made by people. That's actually always what it is. And I don't know how to solve that. So in our case, it was we started it in whatever, 2014 with the trials together with UCSF. And we said we will do it. And we did it. And before the other people woke up, we were already there. So it's often a matter of who is most interested in it, who really wants it, who thinks it's a good thing, who is the innovator, who is driving it. What I find of strange is the concept of born-again theranosticians. That's something that I don't like because that's me too. I can do it too, Train people so that they are competent and it can come from nuclear medicine or radiation oncology. And then there are certain leadership features that make people qualified for a leadership position, then they will run it. So I don't know how to keep it out of politics. That's because people are political. So I don't know.
No, thank you for that. But I want to take a step back with regards to some of the other challenges that we wanted to touch on and turn to Mark with regards to the actual isotope availability and production as a barrier, because you have that as a worldwide horizon on that, as well as Canadian. Talk about that.
Yeah. We are here for this Canadian summit, and I mentioned also in our preparation talk that just doing it for Canada, the population of 40 million is a little bit too small to set up the whole industry and all the infrastructure for that. So I think the radiopharmaceutical community has to be international and to collaborate on an international basis. If you look at the isotope manufacturing, just also historically, the molybdenum that was done here in CNL in Canada, and for decades, and was the largest producer. Of course, Europe with research reactors and also another place of manufacturing was also there. But you have on a global ecosystem just a couple of these manufacturing hubs. And you have a huge infrastructure involved. Yeah, mentioned for molybdenum, the reactor, also now for lutetium or cobalt nuclear power plants. So it's not that you just build it in your backyard. There are some other isotopes that are cyclotron based. Yeah, so that's easier to build a cyclotron in your backyard. As there was a moly crisis, and Canada went along with this tier 24 cyclotrons to the technetium on a cyclotron basis. But I think in general, you have really to make sure you have a global availability of these isotopes. And if you make this globally available, you cannot have hundreds of different isotopes, as physicians and so on would like to have. But you should focus to make them really available. And to have also a global quality standard and a harmonization of the regulations, so it's easier to make it available to all communities.
Great. No, thank you. And I want to move on now, and I've intentionally worded this question a bit to be challenging. And Ting, you're the first up for this. How can we ensure that Canada is not a laggard when it comes to innovation in this area? Now, I've worded that intentionally. I'll use examples, and I hope I don't get booed off the stage, but I'll use examples, proton beam, or I'll use examples, robotic surgery, as late adopters of that. How do we ensure that we remain at the forefront of implementation and translation to our patients in this area?
Well, I think it comes back to your first question, because after all, our healthcare systems is kind of based on our tax dollars. So we have to convince the review boards that whatever we introduce is being cost effective, right? So I think Johannes in his keynote speech has talked about kind of to 50% effectiveness, even. So therefore, it is very important in order to convince those drug review boards that how we could improve this effectiveness. So one particular area, being a medical physicist, that I would be interested in would be, you say, whether we could do personalized dosimetry, and hence we would kind of improve the effectiveness, but then keep the adverse reactions at bay. So that is one of the ways. So there are all this other kind of, again, the imaging, then we have to kind of prove that cancer is heterogeneous. So therefore kind of there is also a way to improve the effectiveness of identifying the right patient, give them the right dose at the right time. So that is the goal of personalized.
Yeah, so Kate, thanks. And so Kate, going from the target at the bench side and the clinical trials, 1 to 3, and then translation into a universal healthcare system like the NHS where you are, how do we ensure that pathway to innovation becomes a highway?
Yes, so I think the first thing to say is that, Canada is not unique in terms of potential problems to implementation of new technology. This is probably a global problem, but certainly a problem in the UK, I'd say. I think, for example, in the UK, there's a recognition that there's a lot of good fundamental research. Certainly at my university, one of the solutions has been to develop a lot of spin-out companies to try and, to speed up the outputs of the research. But there's definitely a problem with introducing new treatments into the NHS. And not least of the problems in the field of radiotheranostics is capacity to treat patients. Suddenly, well, not quite yet in the UK, but there will be a surge of patients with metastatic prostate cancer coming through for this treatment. And until now, most hospital radiopharmacies are quite limited in the number of patients that they can treat. So there's a problem with, capacity, the number of personnel needed to treat these patients. And all of that is being discussed actively in the UK at the moment, but I'm not sure that we have all the solutions by any means at this point. Yeah.
Well, thank you for that. Maybe I'll just quickly 1 minute to Johan and then to Mark. What's the perception? And you're probably going to be polite, but don't worry. This won't go outside this room. But what's the perception of Canada as a innovator in this area or others?
Well, with regards to radioisotopes, it's clearly a leader. That's not a question. With regards to implementing, then I take the fifth. I mean,
you want to do it in that
States. I mean, I find it to a certain degree bizarre that PET imaging is still an issue. I mean, that people would say we don't pay for PET imaging is grotesque. It is bizarre. And I find it also not helpful if one ignores international trial data and says we need to have our own all the time. I find that also bizarre, because why not share data? And they could be publicly available, they could be reviewed, they could be looked at. Sometimes I think it hinges on definitions. What is cost effective? If an 82-year-old patient lives another year, how much is that allowed to cost? And then we're getting to a kind of a very slippery slope in our philosophical and ethical discussions. So, I think less bureaucracy, less people who regulate everything, know everything, tell everybody what to do is usually better for progress, and it's better for patients, I think. Especially if this is known to be completely safe. I mean, there's no question about safety. And if you talk to patients who underwent chemotherapy and subsequently had radiopharmaceutical therapies, they will tell you it's like nothing. So I think it's important to look at these things.
And just to ensure that people don't go away thinking we don't pay for PET in the province, we do for a wide range of indications, but perhaps not as many perhaps as that you might be referring to, but just want to make sure. Mark, same question to you.
Well, Canada being a laggard or being in the forefront, I think we all would not sit here if Canada would be a laggard or behind all this theranostics. So I think Canada is well positioned and really in the forefront. Now talking again from the isotopes perspective. So CNIC was founded several years ago and has brought together a great international audience also. And yeah, me personally being every second month, at least here in Canada, and we have the collaborations with Isogen Bruce Power, but also with CNL on actinium. And it's really Canada where we can move things forward. On the other hand, regulators also tend to collaborate, yeah, and they listen and they're open, and we are very happy. So also for a Type B container fleet and so on, that all goes through Canada. So it's very active, and you have this legacy also, and the nuclear infrastructure that, for example, in Germany is just dying. And so well advanced here. The other thing is we thought also as ITM, where do we put our next production site for lutetium. And we just found some regulations here in Canada that say, well, that's not in the right place, because we have to, it's really considered this lutetium in terms of activities, if you go to 10 to 15 becquerel per year, yeah, to be then like a nuclear power plant in terms of regulations. So this is then, of course, again, a drawback. But overall, definitely in the forefront.
Oh, thanks. Thank you for that. Now, the last question, we got 5 minutes left, so each get 1 minute or so. First of all, to Johan and to Mark, the question is, can you describe, and you really almost have, Johan, so describe a specific success story in which you witnessed an industry, and then to Kate and Ting, the follow-up is, where do you see this success leading the treatment of patients in the future in new areas? So Johan first, then Mark, then Kate, then Ting.
I think everybody knows the industry success stories with Novartis and Endocyte, and Race Bio is now what BMS, and then AstraZeneca is in, and all the major companies are in, except for Pfizer, who announced during ASCO that they will stay away from radiopharmaceutical therapies because it's too complex. So, but everybody else is in there, and that's really a major advantage, because these companies can do major clinical trials. They can look at combination treatments. They can do this, and it has given rise to all these startup companies that have interesting new targets, new imaging approaches, as well as new therapeutic approaches. So I think this is here to stay. And I mentioned that before. There was the late Sam Gambino had talked to about radiotheranostics, and he said in his classic way, well, this is only going to last about 15 to 20 years. And I said, well, good enough for me, So no, I think it's a great success story.
So I think the takeaway there is this is here to stay. Mark?
A personal success story, I mentioned before, I touched that briefly, our success here with Bruce Power really having a nuclear power plant who is willing to install irradiation systems, but OPG also with the cobalt irradiation and so on. And this openness and really to get to a success and make it a success, and also supported by the government. So when we had the first chats with Bruce Power and so on, we suddenly got a phone call from the Ontario office in Munich and said, hey, you tried to do something in Canada, how can we help you and support you? So that's really the right setting, and we feel very welcome here, and that's why we want to continue here in Canada.
So a great message there, great industry, government, and of course, research, clinical relationship, right? Collaboration. Ting.
Okay, so the success story of Novartis has kind of, as Johannes has kind of established radiotheranostics now. So I think the next frontier that we could benefit the patients is the identification of those new targets. And I think we should really push the AI that the federal government now has spent a lot of money on it. So now there is a new development of AlphaFold 3 from Google DeepMind that is out essentially let you do in silico drug discovery. So that would be able to say, for example, going from the monospecific targeting now to the bispecific targeting. For example, identify antigens besides PSMA and other antigens that you could bind to two that would make it much more specific and increase the effectiveness and reduce the adverse reactions.
So message, combination, and where is AI going to play a role in the future? Kate.
So we haven't talked too much about dosimetry, but I think that a success is that the current dogma that all patients should get the same dose, 7.4 gigabecs is good enough for everybody, is being dispelled. And certainly in Europe and the UK, there is now a regulated requirement for patients to have personalized dosimetry, and I regard that as a success.
Great. So this chat, I was about to say panel, but this fireside chat was meant to set the stage for the rest of this 2 days, where a lot of the issues, a lot of the points that have been brought forward by these leaders will continue to be discussed. And most of all, the importance of collaboration, networking, and pushing this field forward. I want to thank the chatters. You saw although not a fire, you certainly saw a passion. And our job is to look to the future, the horizon, and make this happen for our patients. Thanks very much to all of you.
Thank you very much again to our panel. It is now our first coffee and networking break. Refreshments are served outside for 20 minutes, and we will begin.